Hi everyone, this is the Antibiotic Sensitivity lab. In this lab, you're going to learn how to test if bacteria are resistant to different antibiotics or if they're sensitive. So sensitive means that the antibiotic actually works. And I think this is a very cool lab if we were actually doing it because it is actually used in clinical settings when someone has an infection. I'm going to start with background, but just to show this picture, this is what you're going to be ultimately doing, where you take an agar plate and you put bacteria everywhere. We call that a lawn of bacteria. And then you take these different pieces of paper, which we call them discs, and they're dipped in high concentrations of antibiotics. And then you spread them throughout the bacteria and you incubate the plate. And if the bacteria dies around the plate or you don't see any more growth, inhibition of growth, it means that that antibiotic is effective. And we'll talk about it. So we're going to start with just some background on antibiotics. Antibiotics either kill or inhibit the growth of microorganisms or microbes. So they're natural substances made by fungi or bacteria to inhibit the growth, meaning limit the growth or kill microorganisms, usually other bacteria. You may wonder why bacteria would produce antibiotics. So if a bacterium does produce antibiotics, they're producing it to kill other bacteria, not themselves, so that they have more room to grow. And a lot of our antibiotics have come from different fungus. Penicillin and sulfa drugs, so both of these are classes of antibiotics, were the first antibiotics discovered. We have a good amount of antibiotics right now, not a lot, but we have a good amount. And in 1928, Alexander Fleming, who was a scientist, found a mold contaminant on an agar plate. So he was growing Staphylococcus aureus on an agar plate, and the story is he left the plate over the weekend or however long he left it on his lab bench, and when he came back he saw a mold growth, which was penicillin. Penicillin comes from mold, and around this colony he didn't see bacteria growing, so the bacteria had died, so it's like their growth was inhibited. And it was specifically, he was looking at Staphylococcus aureus. So the mold was Penicillium chrysogenum, and he noticed that this mold did inhibit the growth of different bacteria and specifically Staphylococcus aureus. And so this chemical was named penicillin. And penicillin is part of a big family of antibiotics that targets the cell wall in bacteria. So there's many different ways we can discover antibiotics, but that was the first way that antibiotics were discovered. And in the past, antibiotics were believed to be the magic bullet that would wipe out all bacterial infections, because we saw in the beginning that they were excellent drugs, just curing all these deadly bacterial infections. But with time, and as many of you guys know, bacteria are developing resistance to antibiotics. Bacteria can develop resistance to antibiotics, meaning the antibiotic doesn't work any longer, by two ways. Either they gain mutations over time, over many generations, or they acquire resistance plasmids. So if you remember, in the P Blue lab, you actually gave a resistance plasmid to E. coli that gave it the ability to be resistant to ampicillin. So those are the only two ways that bacteria develop resistance to antibiotics. Either they mutate naturally over time, or you mutate them in lab, or they acquire resistance plasmids, whether that's naturally or artificially in lab. And resistance is promoted with the misuse or overuse of antibiotics with people, with animals. So farmers give a lot of antibiotics to animals, and we'll learn about this in lecture later. So if you have a population of bacteria and a few are drug resistant, the antibiotic that you're taking for an infection will kill all the sensitive ones, and then the ones that are resistant will become the dominant type because they'll have room to grow. So this is why we want to be very careful with misusing and overusing antibiotics for infections where it's not necessarily needed. So Alexander Fleming was working with Staph aureus, but now this bacteria has become a major resistant bacteria. So we're already seeing a lot of MRSA and MRSA. MRSA is methicillin resistant Staph aureus, and MRSA is vancomycin resistant Staph aureus. So with both of these, bacteria, you can no longer use these antibiotics anymore. So if someone comes to you with a MRSA infection, which unfortunately is very common these days, we cannot give them antibiotics from this whole big family of drugs. And imagine if one day these bacteria become resistant to all the antibiotics we had. So the rise in antibiotic resistance fueled a need for newer antibiotics and research, but large pharma companies don't want to produce antibiotics because they're expensive, like most research, take lots of time, take lots of effort, and then the fear is that if you're a pharmacy company and you do this, your drug may not be effective in 5-10 years because it develops resistance. So this is a big healthcare problem. And so for doctors, the best thing they can do is to, when they have someone with an infection, to determine the susceptibility or sensitivity of that infection to various antibiotics to give the patient the proper drug and the proper dosage. And when you're a doctor, the best dosage is the minimum dosage that will cure the infection. You don't want to just give these broad range antibiotics for every single infection. We want to be very specific with what we give in a limited amount so that bacteria, the resistant ones, don't overgrow. So doctors send bacterial samples when someone has an infection to labs, and labs can do different antibiotic susceptibility testing, and you're going to be doing one of them in lab. And antibiotic susceptibility testing is the same thing as saying antibiotic sensitivity testing. So you're taking bacteria and you're seeing, is it susceptible or is it sensitive to an antibiotic? And with these, we're trying to determine something called the MIC, which is called the minimum inhibitory concentration. This is very important. This is the minimal amount of a drug concentration that you need to inhibit bacteria. So if you have like Staph aureus, someone who has a Staph aureus infection, MRSA infection, you can give them 100 milligrams of one of the antibiotics and it'll work. But if 10 micrograms works, you'd rather give them the least amount, the minimum amount. So we always try to determine the minimum inhibitory concentration because in the end, antibiotics are chemicals. We don't want bacteria to develop resistance, and they can have some toxic effects with humans as well. So we're going to learn about 3 different susceptibility testing methods. You're going to learn about the broth dilution test, the E-test, and the Kirby Bauer disc diffusion test, which is what you are doing in lab. So you're doing one of the 3, but we're going to learn about all 3 of them. The broth dilution method is you take bacteria and you grow it in different dilutions of an antibiotic. So for example, let's say this is penicillin or ampicillin or polymyxin or tetracycline, whatever antibiotic. You have the antibiotic in different concentrations, from the least concentration to the most concentration, and then you grow bacteria in each of the broth tubes and you see if bacteria grow. So here, this tube is cloudy or it's turbid, meaning there is bacterial growth. It's still growing here, but then look at 8 micrograms of milliliter of whatever antibiotic this is, it looks like we have limited growth. And as you go on, you don't see growth anymore. If given the choice to give this person 8 micrograms per milliliter or 32, yes, both would work, but as a doctor, you would give them the smallest concentration or the MIC. So with the broth dilution method, you can determine the MIC. So that's one test. There are other agar plate sensitivity tests, and the 2 tests you're going to learn about are Kirby Bauer disc diffusion test and the E-test, and both of them use agar plates. So with both tests, what you do is you take an agar plate and you spread bacteria all over. Let's say that someone comes to you with a sore throat. This is an easy example, or maybe like a lung infection or pneumonia. You take a sample, you grow the bacteria in lab, and then you swab it everywhere all over the agar plate. So you put it everywhere, and we call that a lawn. And then on that lawn, with the Kirby Bauer disc diffusion test, you have these small discs. They're like little pieces of paper with different concentrations of antibiotics, and they're placed all around the plate. So if you look at this plate, they're testing 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 different antibiotics, which is a lot to put on one plate, but it's a picture. They're testing different antibiotics. Then you incubate the plate and you come back. Wherever around a disc you don't see growth, that's called growth inhibition, which means that that antibiotic is potentially effective. So With this, you're determining whether the bacteria grew or not, and we call that a zone of inhibition around discs. A zone of inhibition is a good thing because it means that that bacteria, that antibiotic inhibited the growth of the bacteria. And with this, I need to tell you, you cannot determine any concentration. So with the Kirby-Bauer disc diffusion test, we're not determining MIC. We're just seeing, is the bacteria sensitive or resistant? So this is a good first test if you're testing different antibiotics. What I would do is I would test different antibiotics and see which one the bacteria are sensitive to. And then those antibiotics, I would go and do a broth dilution test and dilute them to figure out the minimum concentration. Because with the Kirby-Bauer disc diffusion test, you're not determining any concentrations. The e-test is a great test because you're using antibiotic strips that have different concentrations of antibiotics. So with the e-test, you can determine the minimum inhibitory concentration. And so you look, so this is a bacterial plate with bacteria everywhere. Anywhere you see clearings, these are zones of inhibition where bacteria didn't grow. And there's different concentrations of the antibiotic. The smallest part, the teardrop, is the minimum inhibitory concentration. So now we learned about 3 tests. The broth dilution test, Kirby-Bauer disc diffusion, and e-test. The only 2 that let you determine MIC, or minimum inhibitory concentration, is the broth dilution test and the e-test. The Kirby-Bauer doesn't, but it's a great test to determine sensitivity. And that's the test that you're doing in lab. So in lab, you're gonna test 5 different antibiotics using a Kirby-Bauer disc diffusion test. And you're gonna see if the antibiotics are actually effective against the bacteria. You're gonna, if you were doing it in lab, you would do 2 agar plates. You're gonna do one that's E. coli and one that's Pseudomonas aeruginosa. So you're gonna spread the bacteria everywhere. So one plate will have E. coli everywhere, and one plate will have Pseudomonas aeruginosa everywhere. You basically just put bacteria everywhere. And then you let it sit for a little bit to air dry, and then you take the antibiotics. So in lab, you're gonna test 5 different antibiotics, and they're different discs. You're just gonna gently pat the discs on there, and then you're gonna incubate the plates. So then you incubate the plates so that bacteria have time to grow. And the next day when you come, this is what your plate, an example of what it could look like. So let's say that this is E. coli here, and this student had 5 different antibiotics. Wherever you see clearings, that's the zone of inhibition. So you look here, and that's a good thing. That means that the bacteria is susceptible to this antibiotic or sensitive to this. And you're gonna determine if bacteria are resistant or sensitive to the antibiotics. You have these sheets that have measurements that you're going to put on your agar plate, and it tells you whether, like, okay, if I look at this plate, this antibiotic did not work on the bacteria. I still see growth all over it. These 4 antibiotics potentially worked, but you have standard measurements, these that you put over them. They're like these clear pieces of paper that show you whether, like, you would put this piece of paper over this, and it would tell you whether the antibiotic is sensitive or not. So that's how it works. So if we look at, I zoomed in here, this is an antibiotic, and you can't really see it, but there's bacteria here, and there's a zone of inhibition here. The zone of inhibition is much bigger than the intermediate circle. So that means that this antibiotic, this antibiotic right here is very good for this bacterial infection. And sometimes, let me go back. Sometimes if you focus on a zone of inhibition, you'll see one colony growing, or like a few colonies. If you see colonies growing in the zone of inhibition, that means that they're mutant bugs or mutant bacteria that are resistant to that antibiotic that are growing in there right now. So the goals of this lab is to determine what are the different antibiotic sensitivity testing methods, and we talked about broth dilution test, e-test, and Kirby-Bauer disc diffusion. And the only 2 that you can use to determine the minimum inhibitory concentration are the e-test and the broth dilution test. How do bacteria develop resistance to antibiotics? They can either acquire mutations or resistance plasmids. The basic setup of the test, so what you do again is you swab the bacteria everywhere. So someone comes to you with throat infection and you suspect strep throat and you figure out it's strep throat and now you want to determine what antibiotic to give them, you would take an agar plate and you would spread streptococcus bacteria everywhere, and then you would put discs with different antibiotics, and then you would figure out which antibiotic the bacteria are sensitive to or susceptible to. And so you would look at zones of inhibition as sensitive, intermediate, or resistant. And sometimes you see a colony growing in a zone of inhibition. And if you see a colony growing in a zone of inhibition, that means that that bacteria developed resistance, which is not a good thing. It's not something we want. I hope you guys like this experiment. I mean, I wish we were actually doing it because it's a very common experiment that's done, and hopefully everything makes sense to you guys.